Today, I want to address the problem of biological representation, or how a molecule can become about the relationships between other molecules. To do this requires that we revisit the long history of the relationship between thermodynamics and information that begins with Maxwell's demon and became complicated with the introduction of Claude Shannon's appropriation of the concept of entropy as information. But information entropy and thermodynamic entropy are different uses of the same term and formula. Is this a confusion, or is there a deeper relationship between them? This became further complicated by Schrödinger's introduction of the concept of negentropy and Wiener's description of information as negative entropy. To explore this relationship and avoid some of these confusions, I will focus on the process of interpretation in its most generic sense. That is, the process that assigns reference to information. More explicitly, I will ask, can we provide a precise non-mental notion of biological interpretation? And what sort of process can determine that a particular physical feature is about something extrinsic to that feature, which is of relevance to the process? And keeping in mind that referential information is always about something for some beneficiary. And for something to be a beneficiary, it must be susceptible to degradation or elimination. This risk derives from being far from equilibrium, as in the case of life. And this is what makes thermodynamics relevant. The concept of biological information has unfortunately become confused by a tendency to reduce the process of living to replication and to reduce genetic information to nucleotide sequences. This is consistent with the engineering conception of communication, or as Claude Shannon stated in the introduction to his famous report, the fundamental problem of communication is that of reproducing at one point either exactly or approximately a message selected at another point. This only considers the properties of communication medium and ignores all referential and functional properties. But a pattern by itself is not about anything, and replication does not link molecules to functions. So replication produces mutual information, but not referential information. This begs the question of how a molecule can become information. To investigate how a physical process could come to treat a molecule as information about something, I will employ as simple as possible model system, an autogenic virus. It is a hypothetical, but physically realizable, minimally complex molecular model system. And it's modeled after virus structure. But it exhibits empirically observable physical dispositions that unambiguously will exemplify autonomous, functional, referential, and normative properties. Some of the most significant advances in science have been based on the analysis of simplified model systems. A good model system captures the essential features of the problem without obscuring the critical assumptions in unanalyzed complexities. Examples include Boltzmann's molecule in a box, Maxwell's demon, Bohr's atom, or Turing's machine. A good model system should include no unknown or undescribed processes, ensure that all operations are physically realistic, include no opaque black box-like properties, and provide unambiguous exemplifications of the properties of interest. The model I will use for this purpose is based on a hypothetical non-parasitic virus. A simple virus, like the poliovirus, consists of a container or capsid shell, typically made of protein facets, stuck together to enclose an RNA or DNA molecule, and that when these molecules are inserted into a host cell, they commandeer that cell's systems to make more capsid molecules and more copies of the virus RNA or DNA. Since replication requires parasitism, a virus can be inactivated by damaging its DNA or its RNA. A non-parasitic virus, however, would need to use a different, simpler molecular process to reproduce its part. One candidate for this is reciprocal catalysis. Reciprocal catalysis occurs when one catalytic reaction produces a product that catalyzes a second reaction, which produces a product that catalyzes the first, and so on. And this may involve multiple steps. Here I only show two. In contrast, viral capsids self-assemble, as do cell membranes and microtubules, here shown on the right, and many other complex molecular structures within the cell as well. Because of the way the regular geometries and affinities of these molecules can cause them to associate with one another, they can produce sheets, polyhedrons, or tubes. These two processes, reciprocal catalysis and self-assembly, are chemically complementary to one another because they each tend to produce conditions that are necessary for the other to occur. So reciprocal catalysis, for example, produces high locally asymmetric concentrations of a small number of molecular species, while self-assembly requires persistently high local concentrations of a single species of component molecules. Likewise, self-assembly produces constraint on molecular diffusion, while reciprocal catalysis requires limited diffusion of interdependent catalysts. So each produces the other's boundary constraints. These processes can be coupled. So, when one of the molecular products of a reciprocal catalytic cycle tends to self-assemble into a closed structure, encapsulation of the ensemble of reciprocal catalysts becomes likely. This makes possible self-repair and even self-reproduction, as I'll show in a few minutes. Two forms here are depicted, a polyhedral and tubular form, along with a diagram of the process. An autogenic virus can be called an autogen for short. The logic of simple autogenesis is depicted in this more detailed diagram. Molecule A is modified by catalyst F to produce catalyst C, plus a side product N. And catalyst C modifies substrate molecule D to produce catalyst F and a side product G, which tends to self-assemble into a capsid, and will thereby tend to encapsulate catalysts C and F, and prevent them from diffusing away from one another. Because capsid formation will tend to occur most effectively where reciprocal catalysis occurs, capsids will tend to grow to enclose reciprocal catalysts. This constitutes an autogenic work cycle. Damage that causes a stable capsid to spill its catalytic contents into an environment where there are available substrates will reinitiate reciprocal catalysis and capsule molecule formation. This will repair the damage and reconstitute its stable form. Or it might even produce a second copy of the original from the additional component molecules that are generated in the process. In this way, each of these self-organizing processes produces the extrinsic boundary conditions that the other requires. As a result, these extrinsic constraints become internalized and become constantly available to channel the work necessary to maintain and reproduce these very same constraints. An autogen is therefore self-individuated by this dynamical disposition, and is individuated irrespective of whether it's enclosed or partially dispersed. This creates an intrinsic source of causal dispositions, so that external influences and fixed properties can no longer determine its behavior. Autogens are not only able to self-repair, but because of the cycle from open to closed organization, they will also tend to acquire and exchange molecules with their environment. Molecules incidentally share catalytic interreactivity with autogenic molecules, will tend to be incorporated and replicated, creating variant lineages. Those that don't get incorporated or impede it without being lethal will tend to get crowded out in successive reproductions, because they are not replicated. This provides a capacity to correct error and to evolve. So autogenesis provides what amounts to a constraint production and preservation ratchet. This diagram depicts two cycles of damage and self-repair, in which integrity is temporarily lost, but the intrinsic constraints distributed in colocalized molecules enables the recruitment of energy and new substrates from the environment to reconstitute autogenic integrity. This exemplifies an important interdependency between constraint, work, and information. This exemplifies a circular relationship that Stuart Kaufman and colleagues pointed out in a 2008 paper titled Propagating Organization and Inquiry, in which they say it takes constraints on the release of energy for work to happen, but work for the constraints themselves to come into existence, which leads them to conclude that constraints are information and information is constraint. This circular relationship between constraint and work is exemplified in autogenic self-propagation, and the analogy to viral genetics helps to show why information is constraint, which is preserved from one cycle to the next despite complete substrate replacement. This both provides a record and a source of instruction for organizing the work required to preserve this same capacity. The critical property of constraint is its substrate transferability. This enables constraints to preserve a trace of past instantiations and past work. In other words, referential information. To summarize the argument so far, there are 5 holistic properties that even a simple autogenic system exhibits that are not reducible to the physical-chemical properties of its components, and are instead emergent from the intrinsic dispositions of the whole integrated system. They are: 1. Individuation. It intrinsically maintains an unambiguous self versus non-self distinction. 2. Autonomy. It intrinsically embodies and maintains its own boundary conditions via component processes that reciprocally produce the external boundary conditions for each of the parts. 3. Recursive self-maintenance, a term used by Mark Pickard, because it repairs and replicates the critical boundary conditions that are required to repair and replicate these same critical boundary conditions. Normativity. It's predisposed to produce these results, but this can fail. And it has interpretive capacity. This is because by representing its own boundary conditions in new instantiations, it intrinsically represents its own ontology. From this most basic form of self-representation, 2 canonical complications of interpretive dynamics can be derived. The first I'll show is an example of a higher order interpretive capacity that not only represents itself, as in simple autogenesis, but also features of non-self, that is, its environment. This is exemplified by an autogen that is selectively sensitive. This is possible if, 1, the capsule surface has structures onto which relevant substrate molecules will tend to bind, and if, 2, capsule integrity is made increasingly fragile as the number of bound substrates increases. This will make containment more likely to fail and release catalysts in reproductively supportive conditions. This slightly more complicated autogenic system is diagrammed here with surface structures in which D-type substrates can bind to the capsid surface and weaken its integrity. Notice that substrate binding is mediated by an isometry relationship, similarity between substrate and surface, and containment is made weaker by a correlation relation in which there's a form of contiguity, in which the contact of substrates and surface causes the effect. In this way, autogenic dynamics can effectively assay the environment for its potential to support reproduction. Its change of state from inert to dynamic interprets loss of capsid integrity as information about the suitability of the local environment. In this respect, the number of substrates bound to the autogenic capsid thereby serves as a kind of index of the presence or absence of extrinsic supportive conditions, which provides referential information about the environment for this interpretive system. And it provides normative information about the environment that can potentially benefit the perpetuation of this very same interpretive capacity. A third interpretive complexification can provide something further, referential displacement. This is required to overcome a critical limitation on the evolvability of autogenic interpretation. This arises due to the problem of combinatorial catastrophe. A combinatorial catastrophe can arise when many interdependent molecular interactions contribute to autogenesis. As the number of species that need to interact increases, the number of possible cross-reactions increases geometrically. But only a small fraction of these will be supportive of autogenesis. The proliferation of alternative interaction possibilities will compete with supportive interactions, using up critical components and wasting free energy. This will decrease efficiency and impede reproduction. So simple autogenic systems have limited evolvability. So here's a depiction of multiple molecules interacting and their potential of interactions. And as the number of interacting molecules increases, you can see that the possible number of molecular interactions grows exponentially. Viable autogenic chemistry will therefore necessarily involve only a small fraction of these possible interactions. All others will tend to compete with those that result in autogenic repair and reproduction. This problem quickly makes autogenesis improbable beyond very simple forms. So these non-supportive possible reactions need to be selectively suppressed for more complex autogenesis to evolve. This suppression of all but a tiny fraction of possible chemical reactions is one way to actually look at the function of the template molecules of life, the nucleic acids RNA and DNA. A hint as to why these particular molecules have become the principal information-bearing template molecules of life is provided by their dual roles in cell metabolism. They are both the principal molecules for acquiring, storing, and transporting chemical energy within a cell, and also the building blocks of the information-bearing molecules RNA and DNA. Why this coincidence? This has suggested to me an interesting evolutionary scenario. If another of the side products produced by autocatalytic processes in autogenesis is a molecule like ATP that can acquire and give up energy in the form of phosphate bonds, the availability of this generic free energy could facilitate catalysis, speed it up, make it more efficient, and overcome the need for energetic substrate molecules in its environment to actually drive the reciprocal catalysis. As a result, energy-assisted autogenesis should outcompete and outperform spontaneous autogenesis, and it should be therefore favored by natural selection. This diagrams an autogenic reaction network that generates energy-capturing nucleotide molecules, N, that transfers their energy to the process of catalysis. This will allow a wider variety of potential substrate molecules to become useful. Energetic phosphates would be a source of potential damage, unfortunately, in the inert phase of autogenesis. So in this state, they need to be retained in an inactivated form. Nucleotide molecules can, in fact, be safely inactivated and stored by linking them into a polymer in which phosphates become unexposed. This allows storage for later use via depolymerization of the polymer. Interestingly enough, diverse nucleotides will tend to be linked into polymers of random order if there is nothing else that determines this order. Although the sequence pattern of base pairs will therefore be arbitrary, the specific nucleotide sequence will affect the three-dimensional twist in the shape of the polymer at that location. The availability of a template like this can serve as a surface onto which catalysts can weakly bind. The shape difference caused by this local polynucleotide sequence provides a shape that's an attachment site for catalysts with matching shapes. An extended nucleotide sequence will organize attached catalysts into sequences which will determine their differential spatial correlations with each other. This allows dynamical constraints to be represented as structural constraints. Correlation relationships between catalysts will tend to constrain the probability of particular catalytic interactions. It will increase some and suppress others. This enables transferring the dynamical constraints of autogenesis onto a material substrate that constrains these interactions while maintaining functional and therefore referential continuity. This allows the structural constraints of the template molecule to bias and constrain the interaction probabilities of the catalysts. Nucleotide polymers that match across complementary sequences tend to cause them to form more stable double-stranded complexes, shown here on the right side of the diagram. This, of course, becomes the basis for sequence replication, for example, as we find in DNA. This leads to sequence-specific selection. The template molecule thereby offloads some fraction of system dynamical constraints onto a structure that is not directly incorporated into or modified by the dynamics. Sequences that constrain catalytic interaction probabilities that are closer to the optimal interaction probabilities in the network will be selectively retained because of their higher reproduction and repair rates. The resulting displaced and indirect reference caused by this shift of substrates makes possible the evolution of indefinitely complex interpretive processes. Because it is supported by template structure and not by any catalyst intrinsic interaction tendencies, this can shift the source of interaction constraints from catalyst probabilities to template properties. Since the template is not transformed by chemical reactions, it can serve as a more stable source of memory and instruction, allowing catalysts to be replaced by other kinds of molecules and chemical properties. The result is what I will call referential displacement. This is due to the constraints on functional relationships being partially displaced from dynamical relations to a separate physical structure. The substrate transferability of constraints thereby makes it possible to fractionate the previously holistic function of the autogen. Thus, a molecular template can literally represent the topology of the dynamical network of interactions that functions to represent and reproduce itself. As a result, structural constraints embodied in a part, the template, can represent the holistic constraints of the whole of which it is a part. In this way, molecular structure can serve as a record and a means to instruct the dynamics that reproduces the whole of which it's a part. Because of this part-whole recursion, natural selection can modify an increasingly complex structural record to better fit whole system dynamics to its environmental context. Offloading interaction constraints onto static structures enables that structure to reliably represent those critical constraints irrespective of the potential degrading effects of dynamical self-maintenance. In summary, this exemplifies a three-tiered interpretive logic by which referential and instructional information can be derived and evolved. Simple autogenesis is first, which is entirely determined by holistically embodied isomorphic, that is, self-similar constraints. The second is context-sensitive autogenesis, which is determined by augmenting simple autogenesis by isomorphic surface binding of substrates and their correlated effect on capsid integrity, which can be further displaced by the mediation of a template and the isometric binding of catalysts that determines the physical probabilities of interaction by virtue of their positional correlations. The offloading of interaction constraints onto a physically separate and distinct structure enables the interpretive process to, in effect, reinterpret itself over time. This three-tiered structure of interpretive processes is general. Thus, systematic affordance, in which the information-bearing medium is segregated from the constrained dynamical medium, is made possible by ways of coupling isomorphic and correlative affordances so that they can mediate between these two domains. This provides a bridge for maintaining continuity of information despite discontinuity of substrate. This is formally analogous to the ways that iconic and indexical interpretive relationships at all levels make possible the arbitrarity and systematicity of symbolic systems. Unfortunately, I will not have time to talk about this part of the process. In general, this three-tiered nested hierarchy of interpretive processes enables what can be described as interpretive recursion, making it possible to evolve level upon level of interpretive complexity, in which lower levels of interpretive dynamics generate constraints that are displaced from their substrate and their substrate properties, and as a result can provide an informational scaffold on which higher level interpretive processes can be built, level upon level upon level. This interpretive scaffolding logic is well exemplified by the basic logic of molecular genetics. The so-called genetic code enables the transfer of constraint from one kind of molecular substrate to another. In this way, the sequence properties of DNA become coupled to the three-dimensional interaction properties of proteins via the mediation of isometric and correlative relations among RNA molecules, as shown here. This informational continuity, despite change in molecular substrate, is critical to the open-ended evolvability of organisms. It is also the basis for complex recursive interpretive molecular processes. As demonstrated in the case of template-mediated autogenesis, as we've discussed above, the local nucleotide sequence in a DNA molecule affects the twist of the DNA double helix at that location. This facilitates selective binding of proteins to that surface, but these proteins are therefore able to alter the adjacent gene expression. Thus, protein structure specified by DNA sequences can act to promote, inhibit, or regulate the transcription of other DNA sequences. This is a kind of recursive regulation or recursive interpretive process. This recursive capacity for genes to indirectly regulate the expression of other genes and even whole suites of other genes has made the evolution of complex multicelled bodies possible. Duplication and variation of these so-called regulatory genes allows cells to respond to molecular signals from other cells, and thereby regulate local patterns of cell traits to produce theme and variation logic that makes up body plans. For example, the evolutionary duplication and variation of homeobox genes, shown here, is largely responsible for the anterior-posterior segmental morphology of many animal body plans. So this two-stage displacement, first from genes to proteins, and then from within cell to between cell control of gene expression, is responsible for the theme and variation logic of the very diverse body plans that we see in the world. Functional redundancy of duplicate body parts that results can relax selection on other body parts, in which the accumulation of mutations of the genes that control them will produce variations of their structure and function. Variant forms of body parts will therefore tend to diverge and come to complement the functions of each other, thus initiating selection for their synergistic effects. In this way, interpretive processes can hierarchically complexify with respect to one another. This is enabled by progressive displacement of reference from substrate to substrate and level to level, while maintaining the continuity of interpretive informational functioning of the whole. Interpretive scaffolding like this has thus enabled the evolution of ever more complex forms of representational processes in biology, such as cognition and even language. Thank you.